A very good morning, everyone. My name is Tejasvi Bhandari, and it is my pleasure to be your host this morning. On behalf of the Infosys Science Foundation and Mount Carmel College, I'd like to extend a warm welcome to each one of you gathered here today on a Saturday morning. We at MCC consider it our absolute privilege to be able to host this prestigious Infosys Prize lecture on our campus. As many of you already know, in the 15 years since the inception of the Infosys Prize, it has recognized and honored some of the most brilliant minds in research, inspiring and encouraging future generations of scientists and scholars. We couldn't be happier to be a part of something as impactful and transformative as this, celebrating exceptional contributions to science and knowledge. It is also my pleasure to acknowledge the presence of our Principal Dr. Lekha George and the Dean of Natural and Applied Sciences, Dr. Lakshmi. And I would now like to request both of them to please come forward and present our tokens of appreciation to our distinguished guests. We also have our Registrar of Academics and Registrar of Research. I'd request each of them to present a token of appreciation to our four distinguished guests here. So to Professor Mukund Thattai, Professor of Biochemistry, Biophysics and Bioinformatics at the National Center for Biological Sciences, who is also the recipient of the Infosys Prize 2023 in Physical Sciences. To Bhavana Mehra, General Manager at the Infosys Science Foundation. To Sareen Qasim, Communications and Content Specialist at the Infosys Science Foundation. To Pragati Madhugaria, Communications and Outreach Specialist at the Infosys Science Foundation. It is our pleasure to have you amongst us. Now, to offer you an overview of the Infosys Prize and the distinguished lecture series, it is my pleasure to invite Bhavana Mehra, Head of Infosys Science Foundation, to take the stage. Please join me in welcoming Ms. Mehra.
Thank you so much. Thank you all for showing up on a Saturday morning. As was said earlier, Mounts is also my alma mater, so I'm very happy to be back here. I'm the head of the Infosys Science Foundation, and what we do is recognize scientific research and also the humanities and social sciences, because without that there's going to be no development in the country or in the world. Sans borders. Science results are sans borders. And that's pretty much what the Infosys Prize aims to seek out, reward, recognize. And we have a series of lectures called the Infosys Prize Lectures, where Infosys Prize laureates go out and talk to students, to their own alma maters, to inspire them and get the spirit of scientific thinking, sort of get them to think about things scientifically. It's a process that's not restricted to the sciences, and that's what we're here to do today. So I know that Professor Thattai will have a short film introducing him, and I'd just like to give you a brief about him. He's a professor of a professor of biology and biochemistry at the National Center for Biological Sciences in Bangalore. So without further ado, I would like to hand over to Professor Thattai to talk about his prize-winning work, his Infosys Prize-winning work on the exciting journey of cellular life and evolutionary biology. Thank you.
It's a privilege to be able to do curiosity-driven research. Science is not just about applications. It can also help answer fundamental questions about who we are and where we came from. My work combines physics and phylogenetics to retrace how complex cells evolved over billions of years. On some days, we're solving fun puzzles. On others, we're grappling with really hard problems. Nothing makes sense. One of the best parts of being a scientist is meeting interesting people from different areas, seeing things from their point of view. There's always a lot to learn. Thank you. So while we set up, I think there's a little bit of a hiccup, so we'll sort this out. I was asked to also spend some time after the talk talking about my trajectory of how I ended up becoming a scientist and how I ended up in this field that I work in. So maybe I could just start with the background of how I got here. Okay. Seems to have worked. That was the slide. Whatever you did there was correct. Okay, so I'm actually from Bangalore. Oh, this is not going to let me do one thing or the other. Okay, oh. Yeah. You always expect the unexpected. So I'm from Bangalore. I went to school here. I went to college and graduate school, did my PhD in the US. Both my undergrad and graduate degrees were in physics. And by the time even that I'd reached graduate school to do my PhD, and I was working on problems from statistical mechanics and something called condensed matter theoretical physics, I really had no inkling of what modern biology was really about. And part of the reason for this is when you learn biology in school, at least when I went to school, I think it's still sort of like that today. You learn a lot of facts and you learn all these words and vocabulary, and somehow it's not presented in a way that's exciting, which is really a shame because biology is really today the most exciting scientific field to be in. I think there's no question about that. So by accident, I happened to come across some people doing work in biology in the physics department at MIT, where I was doing a PhD, and I decided to take the leap and join this lab. So I actually joined the lab of a person who had just joined the department, and I was his first student. And our goal was to somehow use methods from physics, which he was trained in and so was I, to try and understand how living cells work. And if you think that sounds very, very strange, in some sense it is, because cells are very squishy and unpredictable, and you don't really think that they would sort of obey the kinds of equations that say planets or electrons obey. But the fact is they do, because they're also physical objects, and we were actually extremely successful in applying methods from physics to understand how cells work. So after I finished my PhD, I moved back to Bangalore in 20 years ago now, in 2004, and I set up a lab at this place, the National Center for Biological Sciences, where I continue to work. And as soon as this starts to work, I'll be happy to share some of this work with you. Before we start, can I just get a snapshot of who's in the audience here? How many of you are students of biology or biochemistry? Could you just raise your hands? It's about half. And how many from, say, physics or math? There's a few over here. Thank you. Engineering? No engineering here. Yeah, but there may be people from around town. Okay. Good. And how many of you are UG students? Wow. Okay. And how many PG? Half and half. Okay. Very good. Fine. So I think I'll pitch this to the right level. After the talk is over, we'll have time for questions. And if there's anything that's not clear during the talk, please feel free to interrupt and ask. Okay, so I'm just going to run back and forth all over the stage. Okay, good. So like I said, I came from physics, but now I'm doing biology. And you have to have some way to use the tools and the skills that you were trained with when you enter a different field. And one of the questions that in some sense anchors how I approach biology is this question, which all of you, hopefully have thought about. It's certainly something that people have been wondering about for a long time. if, surely there's life on other planets, that must be the case, but what would it look like, right? If we manage to actually go there and probe what life looked like, not on Mars, on some very, very distant planet, orbiting a very, very different kind of star, with all kinds of different chemicals than we have here on Earth, would it look the same as life on Earth? How would it look the same and how would it look different? And if you want to organize the concepts of life, the most central one, I think, almost by definition, is the idea of evolution. So as far as we understand, life is inseparable from evolution. Evolution is the mechanism by which things become more complex over time, Darwinian evolution. And I think no matter where life arises, a mechanism like Darwinian evolution must be operating. So it's not something that is sort of negotiable. But what about other aspects of life on Earth? One of the most important things that we discovered about life in the middle of the previous century was the idea that life was, in some sense, completely digital. Right? The idea that there's the genome, and there's the DNA sequence, and there's these letters ATGC, and it's almost like a computer file that's running inside a cell, a program that's being executed to make sure that the offspring, the daughter cell, looks like the parent, or that our offspring look like us, right? That mechanism of heredity. It didn't have to be digital, but it is. And there's sort of very good reasons to expect that no matter where life arises, this very robust way of making a copy of the genetic material in a digital sense and passing it on to the offspring is very likely to be some ingredient of life no matter where we find it. So these two, I think, we can expect. But what about this third aspect, the idea of cells? So I'm going to talk a lot about cells today. But if we went to some other planet and we looked at living things, would they be made of cells? That's not expected at all, right? Because that may be very specific to the history of life here on Earth, right? But if you think about it, what is a cell? A cell is a boundary, it's a membrane, and the membrane separates the outside from the inside. It separates self from non-self. And it turns out that unless you have a separation like this, then evolution as we understand it is not going to work. So I'll quickly explain why. So you know that there's DNA inside the cell, this DNA encodes genes, and the way evolution works is that there are mutations in the genes, and genes that help the organism function better will tend to persist because that organism will have more offspring. If the positive benefits of having a really cool new gene could be shared by a whole bunch of different organisms, and not just selfishly by this particular cell, then evolution wouldn't really work out because all your innovations are being shared in a more widespread way. So the idea of competition, which is the driver of evolutionary complexity, doesn't really work unless you have a boundary between self and non-self. And on Earth, that boundary works as a cell. Somewhere else, it may work as some other sort of physical object, but that cell-like idea is absolutely central. So when I started getting into biology, I realized very quickly that this scale of biology I wanted to work in was the scale of the cell. Whereas there are other people, for example, who are interested in studying the brain or studying ecosystems, and there's many, many different things you could study in biology. So where I work is at the level of the cell, and this is why. So here I'm going to tell you a really amazing story. It's a story that's still ongoing. Science never stops, but it's really quite amazing how recent discoveries have changed the way we think about the origin and evolution of life on Earth. And I'm going to tell you the story of what I call complex cells, maybe with a little bit of a biased view. These are the cells that we call the eukaryotes, which we are an example of those. How did they arise on Earth? What were the mechanisms that caused them to come into being, and how do they continue to evolve as time progresses? So the most central discovery in biology is this one. The idea that all life, every single living thing on Earth, is made of cells. Okay? It wasn't always clear that this was true, but it's absolutely certain now. How did people discover this? By just looking with the naked eye, you would never make a discovery like that. So it had to wait for the development of optical microscopes that allowed people to probe the organization of living matter at very, very small scales that are not visible to the naked eye, at scales of microns, right? The scales of the wavelength of light. Two pioneers in this area were contemporaries, Van Leeuwenhoek on the left and Robert Hooke on the right. Both lived at the same time, both were experts on microscopy and used microscopes to study what the organization of living matter was at the smallest scales. Van Leeuwenhoek would just pick up water, ditch water or pond water and look and put it under the microscope, and he discovered that even sort of transparent water was teeming with life when you looked at it microscopically. It was just literally chock full of organisms. And he made some absolutely beautiful drawings which are comparable to the kinds of images you would get today from a microscope. Okay, but by hand, he would go to his book and draw, go back and look, and we now know, because his drawings are so accurate, that he was able to sample all kinds of cells that were then later discovered and characterized, which represent both eukaryotes and prokaryotes. And he realized that these little objects were sort of fundamental units of life. He called them animalcules. They were just very, very tiny animals, because they moved around, they chased after each other, they had agency, just like all living things. Robert Hooke was looking at a different scale. He was looking at plant tissues, and when he sliced the tissues and put them under the microscope, he realized it wasn't just a sort of continuous mass, but it was broken up into these little subdomains, which he called cells. And he called them cells because they look like the cells of a monastery, right? A room. And that's where the word cell comes from. And between the two of them, it sort of became clear that even a tree, this huge tree, was made up of exactly the kind of building blocks as the unicellular organisms. And this was an astonishing discovery. It's sort of not something you would have guessed just by looking at the way we are organized or trees are organized. That we're made up of these very, very specific building blocks that at the very small scale are identical to free living cells. So, we are like a bunch of free living cells that came together. It's an astonishing idea. So once that became clear that all living things were, had this fundamental unity, the next question, and biology is quite obsessed with this, the idea is the idea of classification. We know all life is cells, but can we then sub-organize life into different domains, or different kingdoms, and then sub-organize them into more related families? Ultimately, this leads to the idea of species and their relationships. But what are the, not looking at a fine scale of the differences between two nearby species, what are the biggest scales of organization in biology? And what people thought in the early days of cell theory was that there were two kinds of organisms, unicellular and multicellular. And this sort of makes sense, right? Trees and blue whales and humans on one side, and E. coli and amoeba and paramecium on the other side. It seems like a very natural way to organize this. But this turns out, again in biology, the obvious thing turns out to be not correct, right? So the big divide, the big evolutionary divide is not unicellular and multicellular, by which I mean that all the multicellular organisms are not really closely related to each other. They all became multicellular separately. And what's really important is to understand their unicellular ancestors that they came from. So the real divide in biology, and I know you'll be yawning when I start to use these words, but is the divide between prokaryotes and eukaryotes. Now you can go and ask your parents or grandparents if they learned about prokaryotes and eukaryotes in the textbooks, and given the age group here, potentially your parents did, but your grandparents may have not, because this definition only really reached the textbooks in the 1970s. Okay, so before, you would think this is like the oldest fact in biology, right? Prokaryotes and eukaryotes, but this is not the case. And the reason it's not the case is nobody could have even guessed what a eukaryote was, because it's defined by its microscopic structure, what's called the ultrastructure. And just like Van Leeuwenhoek and Robert Hooke made their discovery with optical microscopes, the discovery of eukaryotes could not have happened without the electron microscope. And clearly the electron microscope only became something that was a tool that could be used to study living samples in the middle of the 20th century. Right? So it's really a very new fact that there are these things called prokaryotes and things called eukaryotes, and that's how biology is organized. And something even more astonishing. So I just want to read this this paper. This is a very famous paper by Stanier and Van Niel, written in 1961, not that long ago in the scheme of things. And this is a paper that's grappling with the idea in the 1960s, what is a bacterium? Because they had no definition of it in the 1960s. So the introduction is beautiful, and I'm going to read it. So they say, since the earliest days of microbiology, remember microbiology is a very, very old field, the biological nature and relationships of the bacteria have been subjects of perennial discussion. Why have these questions obsessed some members of each succeeding generation of microbiologists? There can be no doubt about the principal reason. Any good biologist finds it intellectually distressing to devote his life or her life to the study of a group that cannot readily and satisfactorily be defined in biological terms. And the abiding intellectual scandal of bacteriology has been the absence of a clear concept of a bacterium. So they're saying that this is crazy. We claim to be doing science. I mean, you do science, you have definitions. And certainly by the 1960s, we had pretty much worked out, quantum mechanics, right? But we hadn't figured out what a bacterium was. And so they say we need definitions. Just like physics needs definitions, biology needs definitions which are rigorous and easy to check. And the definition they gave of what a bacterium was very, very interesting. So they say, Stanley and Van Niel, this is a review of their paper which is much more recent, still could do little more than define the prokaryotes in negative terms from the eukaryotes. So they said, what is a bacterium? It's a non-eukaryote. Which if you think about it, these days you would say, the eukaryote seems to be the thing that should be defined second, because the bacteria are simpler and that should be defined first. I mean, this is like saying, I can't even imagine, right? It's splitting the whole world into left-handed violinists and everybody else, right? It's a very, very funny way to organize things. What are eukaryotes? Well, when you put them under an electron microscope, you find that eukaryotes have a membrane-bound nucleus, a cytoskeleton, an intricate system, of internal membranes, mitochondria that perform respiration, and in the case of plants, chloroplast. So they're listing all the things that you see in every eukaryotic cell, again and again, no matter whether they're unicellular or multicellular. Bacteria, prokaryotes, were smaller and lacked all these structures, they lacked mitosis. So that was the definition of the bacteria, the anti-eukaryote. And you all know, because it's in the textbooks, they're called eukaryotes because they have a nucleus, right? But that's not really why they call it, that's just the name. They're called eukaryotes because they have all these different things. They're huge, they have these floppy membranes, they have these internal organelles, they have the cytoskeleton, they have all these different aspects, right? So the eukaryote is really defined by its complex internal structure. Okay, we have some background music. So then the next question becomes, fine, there are prokaryotes and eukaryotes. So what's the connection between them? And is that the end of the story? So you flash forward just 10 years to the 1970s. And this beautiful image is of a person named Carl Woese, a very modern biologist whose work in the 1970s could well have been done even today. What he decided to do was he said, there's no point looking at systems and subjectively classifying them. One person could say these two cells look similar, another person could say they look different, and then they can't even decide how to organize it. So he said, since now we have the ability to sequence DNA, let's just do it mathematically. So he went around sampling living organisms from as many environments as he could. He extracted a particular gene, which is a ribosomal RNA. It's a gene that all living organisms have, and got the sequence of it. And once you have a sequence, which is a bunch of letters, then you can use mathematics to see if two sequences are closer and two sequences are further apart. And in this way, you can rigorously, objectively in some sense, classify which organisms are more close, which organisms are further apart, and what are the big domains of life. And suddenly, from out of his sequencing machine, a huge surprise jumps up. He was expecting two kinds of organisms, prokaryotes and eukaryotes. But suddenly there are three. And these three organisms are as distant from each other as is possible to imagine. So he split the prokaryotes, previously they were thought to be just bacteria, into two huge groups, which he called archaea and bacteria. And then there was the third wing, which are called the eukaryotes. I don't know if you can see this beautiful tree over here, even the background light, but this is called a phylogenetic tree, and you should think of it as demonstrating the relationships of organisms evolving over time. So there's the root of the tree, and over time the organisms diversify and break into different groups. And this particular tree has three big sort of lobes: the bacteria, the archaea, and the eukaryotes. And the amazing thing is this discovery has since not been turned over. This is the true classification of life on Earth. As far as we have discovered, and unless we discover a totally new kingdom, every organism we know truly falls into these three groups: the bacteria, the archaea, and the eukaryotes. And you might think that's the end of it, but then of course you start to ask the next question. If there are three groups, how are they related to each other? Two of them must have been closer and one of them must have been more distant, right? Because biology evolves by splitting and radiating. So which of these two groups are more closely related? Well, clearly bacteria and archaea are the closest. But where did the eukaryotes come from then? Did they come from the bacteria? Did they come from the archaea? What's the story? And this is the place where things really get turned on their heads. A completely unexpected thing happens. Just to flash forward. So these are some very recent papers that I pulled up from 2019 and 2020, representing, again, using the same kinds of techniques that Carl Woese used, but now applied to literally every organism whose sequence we have. And the tree that Carl Woese wrote, of course, has become more diversified as more and more groups have been added, but the overall topology has not changed. And roughly what we have is, this is the tree of all prokaryotes,
Here you have the bacteria, here you have the archaea, and somehow appearing out of nowhere in some sense, here you have the tree of eukaryotes. The tree of eukaryotes is still work in progress. We don't really know how much we're missing, because a lot of the eukaryotes that are used to construct these trees were discovered in the last 10 years. Right? The bulk of eukaryotic groups that we know about today were discovered in the last 10 years. The groups that we know very well about, one are called the opisthokonts, this one. That little group of this whole tree contains humans, contains yeast, contains all animals and all fungi, and literally everything you learn about in school is in this little branch here. Okay? And nobody learns about any of this other stuff. And there's one other branch which is important, which are the plants. Okay, so when you learn biology, you learn about plants, animals, and protists, and that's totally the wrong way to think about it. Because plants, animals are here, and protists is everything else. Okay. But nevertheless, all eukaryotes truly are related to each other. And the question is where they came from with respect to the other kingdoms. So here's the answer. I'm just, spoiler alert, as they say. So I'm just jumping ahead. Eukaryotes didn't come from archaea. Eukaryotes didn't come from bacteria. They came from both. Okay, and this is a very mysterious thing. How can it be that you can't say that elephants came from two different, from mice and from cows or something like that, right? Biology doesn't work like that. But somehow eukaryotes came from the other two great kingdoms of life, the bacteria and the archaea. So the question is how? And how would somebody even prove something like this, right? So a lot of the legwork for this hypothesis was done by this person. Her name is Lynn Margulis. This is a photo of Lynn Margulis on her wedding day when she took the name Lynn Sagan. And this is Carl Sagan. And if you know Carl Sagan, he was an astronomer and science popularizer. He's part of the reason why I got into science. So Lynn Margulis, like Carl Sagan, was interested in the origin of life and evolution. And she realized something. That when you look inside a eukaryotic cell, one of the standard things that we learned that all eukaryotes have, apart from the nucleus, is the mitochondria. You all know about it, right? Almost all eukaryotes have a mitochondria. There's one or two that don't. And what she realized were mitochondria are not an intrinsic part of the eukaryotic cell, but they seem to be an independent organism that's living inside a eukaryotic cell. It's like a symbiotic relationship, but living inside. And so she called it an endosymbiosis. She realized that mitochondria, at one time, were actually bacteria that lived on their own. And if this sounds strange, actually there's a huge amount of evidence for this. The most obvious piece of evidence for this is that when you stain for DNA inside a cell, and you can just do this experiment in the lab over here, you find that there's DNA in the nucleus, which is this blue stuff, that's our nuclear DNA, but there's also DNA scattered in other parts of the cell, this red stuff. It's mitochondrial DNA. So for some reason, mitochondria came with their own DNA. And of course, now we know very clearly why that is. And this DNA is inherited maternally. There's actually a huge amount of very interesting biology related to mitochondria that people are working on today. But the one thing I want you to understand from this is that mitochondria used to be bacteria. And they ended up living inside another cell. Okay? And became mitochondria. So then the next obvious question is, so what was that other cell? How does one cell go live inside the other? Can we find out what that other cell looked like billions of years ago, whenever this happened? So now, with very modern data, and this I'm really showing data from 2023, what people have done is to find a hint of what the other cell was. It's a very straightforward procedure. You take all the genes in a eukaryotic organism, like you take all human genes, we have some 20-something thousand genes, and each gene, you look at it, you squint, and you say, does it look like a bacterial gene, or does it look like some other kind of gene? And you try and map it back to a prokaryotic ancestor. And usually when you do this, you would expect it all maps back to one ancestral type of organism. But what you end up finding is all the eukaryotic genes, half of them, roughly, map to bacteria, and half of them map to archaea. That means your nucleus is full of genes, some of which come from bacteria, and some of which come from archaea, and all of which are working together. Right? So this is a crazy thing. And it's also very systematic. All your genes that are responsible for DNA replication, transcription, translation, come from archaea. All your genes that are responsible for metabolism and certain kinds of chemistry, they come from bacteria. And in the same way, there's a very specific functional split up of how many of your genes come from bacteria, how many come from archaea. So it really seems to suggest that if mitochondria were bacteria that lived on their own, the host cell must have been an archaea, the other type of cell. And this also matches with the timeline. So people have now done fossil studies of eukaryotes and fossil studies of all cells. You find these rocks where you can thin slice them, send them to the electron microscope, also do mass spectrometry and identify chemical compositions. So it turns out that the eukaryotes are relatively recent, right? And at least 2 billion years old, and before that you only have prokaryotic type of cells. So it really seems to say that on Earth, from 3.5 billion years ago, when life first arose, to 2 billion years ago, there were only prokaryotes, both archaea and bacteria. Suddenly, 2 billion years ago, give or take, there's this new type of organism, the eukaryote, which seems to be a mix of both. Okay, so again, electron microscopy is used to look at the history of the eukaryotic fossil record, and you find that as early as 1.6 billion years ago, you find cells in the fossil record which, just like Stanier and Van Niel found in the 1970s, these are clearly non-prokaryotic cells. They're much bigger. They have this wonderful internal structure, right? I just want you to stare at this for a few seconds. These are electron micrographs of cells that are 1.6 billion years old. So I met the person who does this. She goes around, her name is Emmanuel Javo. She goes around looking for rocks, slicing them, putting them in a microscope. 99.999% of the time there's nothing over there. But occasionally you find, because these cells are very small and difficult to fossilize. They're not like dinosaur bones, right? Okay, so we know that mitochondria were bacteria. In fact, we know what kind of bacteria they were. They're called alpha proteobacteria, just like E. coli, which some of you may have studied in the lab, right? So mitochondria are cousins of E. coli, and they're not very mysterious. But what was the host cell? It was archaeal, but we couldn't really put our finger on what it was. And so the question was, well, maybe the archaea that eukaryotes came from were already complex. And somehow the complexity of eukaryotes is a much older problem. Or it could be that the archaea that eukaryotes came from was simple, and that there's some subsequent leap of complexity that happened later. So which one of these two things? To answer that question, you have to find the closest relative of the archaeal host living today. And the answer for this comes from Asgard, okay? Which of course I know all of you are familiar with, yeah? But it's not these guys. With apologies to Marvel, no. So it comes from this place. This is a deep sea vent off the coast of Norway, and it's called Loki's Castle. It's about 2.5 kilometers below the surface. And in the just before 2010, a submarine sampling experiment went and got all the sediments from here, brought it up, and sequenced it. And the problem is when you bring cells up from 2.5 kilometers below the surface of the Earth, by the time they're used to living under pressure. So by the time you bring them up, they can't live at ordinary pressure. It's very difficult to actually bring them up and culture them. But you can get their DNA. And so what happened was they sequenced the DNA from here. And suddenly, just popping out of this, what they found was clearly an archaeal genome. It's a genome that looks just like you expect for an archaea, which remember is a prokaryote. But the genes of those archaea look very similar to our genes, eukaryotes. So they said this must have been the present day cousin of the host cell that 2 billion years ago would have been the host for the eukaryogenesis event. Okay? And this is the most recent tree from 2023. This is the tree of archaea, but not all archaea. One group of archaea, which because of Loki's castle are named the Asgard superphylum of archaea. And inside there you have Loki archaea and Thor archaea and so on. And here you have Heimdall archaea. And the Heimdall archaea turn out to be the closest relatives of the eukaryotes. Okay? So, very, very nice names, very good for marketing. But it turns out that actually this group of archaea are not confined to that deep sea vent. You can actually find them everywhere. They've now been found on every continent except Antarctica. They live in certain kinds of sediments. So we didn't come from the deep sea vent, that's not what I'm saying. But we came from this particular group of archaea. So the next question is, what do those cells look like? And are they already complex? Now it turns out that these cells were very difficult to culture. And after a 10-year effort, a group in Japan actually managed to culture them. And this is what those cells look like. So they're very small. This is 1 micron. And this is what a Loki archaeota looks like. This is the kind of cell that they would have found in the deep sea vent. They are interesting. I mean, they have these protrusions and they have something called a cytoskeleton, which eukaryotes also have. But overall, they don't look as complex as a eukaryote, right? So somehow, even if these were the host cells, there must have been a leap
that happen in complexity that we still have to explain.
In contrast, if you look at the structure of a typical eukaryote, first of all, they're much bigger. This will be maybe 50 nanometers, so this is a very high magnification ultrastructure. This is a beautiful electron micrograph by a person named George Palladi, a Nobel Prize winner, from the 1980s. The inside of a eukaryotic cell is absolutely intricate and complex. Here's the nucleus, the Golgi apparatus, there are vesicles or vacuoles, there are small transport vesicles. There's all kinds of things inside a eukaryotic cell that you just don't find if you look inside a standard bacterium or a standard archaeon, even the archaea that we came from. So somehow the combination of two simple cells ends up making a complex cell. So the question is, how does that happen? So here I want to pause and just go back to this original thing I said about Darwinian evolution. So in biology, when you are asked, how does biological diversity arise? How does biological complexity arise? The answer is always Darwinian evolution, and the key to Darwinian evolution is competition. That is, there's some limited resources, there are a bunch of organisms competing for those, and the one that does better has more offspring. That's survival of the fittest, right? So this kind of brutish competition is the origin of biological complexity. But here, the single biggest change in biology since the origin of life, instead of competition, what do you have? You have cooperation. You have somehow two subgroups of organisms which come together and make something that's greater than either piece on its own. And so the idea of cooperation in biology is actually grossly understudied. And it turns out to be a very, very important part about how biological complexity arises. Maybe it's not surprising. There's lots of symbiotic relationships, and it's just surprising that people haven't spent much time quantifying all that. It's really an open question. How much competition and how much cooperation influences the long-term evolution of life on Earth? So, fine. What's the story? So at this point, we can only make up, everything I'm giving you are facts, verifiable, but at this point, then you have to just make up stories. So here's the example of a story of how a simple cell or a combination of simple cells could have become complex. This is a story that's put forward by my friend Buzz a few years ago, and I actually buy into this particular hypothesis. The idea is the following. The reason it took 10 years to culture this organism is because if you just culture that organism on its own, it doesn't survive. If you put into that medium any amount of goodies, sugar, salt, and all that, that's not enough. It turns out that these types of archaea need bacteria in the liquid in order to survive. And that's because they are already in a symbiotic relationship with bacteria. So this is interesting, right? And how does that work? Typically, there are bacteria on the surface of these cells, and the archaea make some chemicals which the bacteria absorb. The bacteria make some chemicals which the archaea absorb, and by exchanging these chemicals, they both have a metabolic symbiosis. They both need each other. It's called mutualism. So this is the clue, right? So it turns out that these Loki archaea, or all the Asgard archaea, they have these long tendrils, which makes a big surface area, and there are bacteria that sit in the pockets of those tendrils, and they grow as a combined multi-species unit. Okay, so the idea was that over the course of hundreds of millions of years, the tight fit between these two membranes becomes ever more entwined until you have almost an inability to distinguish what is the inside, what is the outside. The bacteria end up being deep inside the cell. They become the mitochondria. The original cell becomes the nucleus, and then you have the rest of the cell, which is now, because it has this energy surplus of mitochondria, much bigger. The genomes are 1,000 times bigger. They can do a lot more cool things, and you get a eukaryote. This is different from what they say in the textbooks. If you read the textbooks, you see these crazy pictures where you have one cell, you have another cell, it goes inside, and suddenly you have a eukaryote. And biology doesn't work like that. It doesn't work by one-off events. It works by these long, slow, population level changes. Okay, so this was the background. So I thought I would take a little bit of time to actually also describe the work that we do in the group, so you can get a snapshot of how a physicist is able to approach biology. So I'm going to take the next 10 minutes or so to talk about that. So we are interested in eukaryotic evolution, but when you do science, you can't pick such a big topic. You have to pick something specific. So we're interested in a specific subsystem inside every eukaryote, which is called the vesicle trafficking apparatus. And I'm interested in its origin and evolution. If you've never heard about it, don't worry. I hadn't either until some time ago. So I'm going to tell you what it is. So here's the work that we've done over the past 15 years or so. So this is the cartoon that I showed you, but drawn in a slightly more stylized way. So these the original proto-eukaryote, the population from which proto-eukaryotes came. These are the archaea, these are the bacteria. They were in a symbiosis, but not an endosymbiosis. The mitochondria go inside, you end up getting a nucleus. Then over the course of time, somehow the cell becomes bigger and more complex and has more genes. This is a billion-year branch. And the problem with that branch is that all the side lineages from that branch, which must have been alive on Earth sometime, have all gone extinct. This tends to happen in biology, right? As lineages go extinct, you lose information about the past. Human evolution, for example, we have no extant cousins of humans that bridge the gap between the chimpanzee and human common ancestor. Although fossil record shows that at some point on Earth, there were a large number of hominin species, many even living at the same time. So in the same way, there are lots of creatures that span the first eukaryote to the last eukaryote, where all these new features, the nucleus, the cytoskeleton, mitosis, all this cool stuff came about one by one, but we can't retrace that because all those missing links died out. Instead, you get this, which is the ancestor of all living eukaryotes, which then diversify into these amazing protist and multicellular groups that we see today. So the work we've done in the group spans this entire sort of 1.5 billion year canvas. We've worked on the archaeal origins of eukaryotes, on the symbiosis of mitochondria, on the origins of the first membranes, and on the origins of new organelles, including my favorite organelle, which is called the Golgi apparatus. And I'll give you a few vignettes from many different studies that we've done that shed light on how we do these things. So one question. Remember I said that when you bring two cells together, the combination is greater than the individual parts. So how is it that the eukaryote is able to do things that neither the archaea nor the bacteria were able to do? We can't answer this question in general, but we actually discovered one cool thing. Inside the eukaryote, there are all these membranes. Imagine sitting inside the cell, there's all these different surfaces. How does a molecule inside the cell know which surface it's going towards? If they all had different colors or labels, that makes it easier, right? So it turns out that in a eukaryotic cell, the membranes are quite distinguishable. They are chemically very different because they have these labels, which are this molecule called the Rab GTPase, it doesn't matter. But there are dozens of kinds of Rab GTPases. You can think of them as different colors, and they color the different membranes differently. So any molecule in the cell knows where to go. The Rab GTPase has two pieces. It has one bit which is the color part, the other bit which is the membrane insertion part. It turns out that the color part comes from archaea, and the membrane insertion part comes from bacteria, and neither group on its own has an ability to decorate membranes, but when you put them together, you have the ability to do that. So this is an example of how combining capabilities from two different groups gives you something completely new. We also studied how mitochondria were engulfed. Remember mitochondria are bacteria. What do bacteria like to do? They like to grow and divide. That's what they like to do. So if you put a bacterium inside another cell, typically what happens is it's a pathogen. Right? It happens, our cells are also full of bacteria. We get infected all the time. So what does it do? It grows and grows and grows and destroys the host cell. So how will the host cell control the symbiont? Right? Not just by goodwill, right? So the way it happens is that bacteria have a way to divide by pinching their membranes from the inside. And they can divide rather rapidly. And they divide using a system called FTSZ, which is an internal little loop that pinches the bacterial membrane. In our cells, the mitochondria don't divide like that. Instead, what happens is the host cell sends a ring from the outside to pinch the mitochondria. And this is how it tames the growth. If the mitochondria are growing too rapidly, then the division rate is slowed down. And so there's a balance between mitochondrial division and host cell division. So we did some work to understand how could it be that you transition from this mechanism of internal division to a mechanism of external division. And we discovered something very interesting, that there are actually a few subgroups of eukaryotes today, some amoeba, some rarely, and some things called stramenopiles with the diatoms, where you can show that the mitochondria look exactly like they looked 2 billion years ago because they divide in the bacterial way. So these are living fossils. These are examples of an ancient mechanism, 2 billion year old, which is still persistent in some organisms, but not in others. And in some sense, if you want to study the history of eukaryotes, you should go and look at these funny groups. Now, the reason this was not discovered earlier is something which is absolutely, I think putting biology on the wrong track. When people study organisms today, they tend to study the organisms which other people have already studied. You study yeast, or you study fruit flies, or you study Arabidopsis, right? You study these model organisms. And model organisms are not representative of the diversity of eukaryotes. And so the pitch to you guys is, you should just pick up any organism you want that's interesting and go study it. Okay, We've also done some work on the idea that eukaryote genomes are 1,000 times bigger in terms of amount of DNA and have several fold more genes. So the question is, somehow having more genes allows eukaryotes to be complex. How does that work? And one of the places where that seems to be very important is in the vesicle trafficking system. So to wrap up, I'm going to give you some examples of the work we've done to model the vesicle traffic system and look at its evolution. The fact is that a lot of this work, as I said, is impossible to do directly because there are no fossils, there are no intermediate forms. So how do you go back in time if you don't have an actual organism to study? And so what we do is we make models using as much information as possible about how we know cells work today. And then using those models, we crank back the proteins to what we expect was their ancestral state 2 billion years ago, and we see what things look like, and then run the tape of evolution again to see what happens. Right? So I'm going to give you a snapshot of how this works. So here's what a vesicle trafficking system is. You've already seen a little bit of this. So here you have, this is a big cell, here you have nucleus, the ER, the Golgi, the endosomes, the plasma membrane. When you see a snapshot like this in a textbook, it doesn't give you a picture of how dynamic the whole thing is. Okay? So this is not just a God-given template. It's not like a cookie cutter where you just make it and then it just stays like that. Everything exchanges stuff. Okay? It's like having warehouses all over the city, and you have these Amazon trucks that are moving stuff between the different warehouses. So there are chemicals being exchanged between every one of these warehouses. If that's happening, how does the composition of each warehouse stay the same? Right? It's very, very unexpected. Somehow the cell stays the same even though there's so much dynamism inside. And that's really the key to understanding the vesicle traffic system. It's called something which goes by the name of emergence in physics or math. The idea that the structure actually emerges from extremely dynamic microscopic interactions. The key to that whole thing is this object. It's about 40 nanometers. It's called a transport vesicle. This is the transporter that moves cargo from one place to the other. So this is the truck that's moving things between different warehouses. The cargo is on the inside, potentially on the surface. And how does this vesicle know where to go? It's because of all these amazing molecules on the surface that are specific to different locations, right? In a lock and key kind of way. This is how that works. If you have a source compartment, like ER, and you have a target compartment like the Golgi, what happens is you make a vesicle from here, and this vesicle is made by a very specific kind of coat, which will take only certain cargo. You go to the warehouse and say, no, I'm only transporting, the TV or whatever it is from that warehouse. Once you package it, the vesicle then moves around in the cell, finds a place to fuse and deposit the cargo. How does it know where to fuse? Not because anybody else is telling it anything. It's because some of the molecules on the surface are actually responsible in a lock and key way for finding a partner molecule on the target membrane and fusing it. These are called snares. And just like I said, there are many different kinds of rad GTPases that color different membranes. There are many kinds of snares. So each vesicle with extremely high fidelity will only fuse to the correct target. It will deliver exactly where it needs to deliver. So this is what's happening at the microscopic level. But at the macroscopic level, like I said, there is this funny idea. Even though things are moving around, you're not changing the composition of every compartment. So this then becomes a purely mathematical problem. And so over the years, we've actually made several kinds of models spanning the scale from genes to proteins to networks to function to understand how all this works. For those of you in the audience who've done a bit of math, it turns out you can use differential equations in dynamical systems. You can use things called Boolean stochastic networks. You can even use something called graph theory, which is a very cool and very powerful group of tools in mathematics to understand how things move around. Graph theory was actually used in the 70s to understand how traffic moves around in a city. And we use exactly the same theorems to understand how cells move cargo around a cell. That's why it's called vesicle traffic. And it all seems very complicated, but I'm going to leave you with the model we're actually working on today, which turns out to be something you can just do on pen and paper. You don't need to know any math to do these kinds of models. Okay. So here's an example of a model. Remember I said there are these coats? The coat is what selects the right cargo. So imagine there are 3 different kinds of coats in the cell, and the different kinds of cargo will either like or not like some coat. So how many different kinds of cargo can there be? It can be 2 to the 3, which is 8. If the coats are colored red, green, and blue, then depending on which coats the cargo like, for example, this cargo likes both red and blue, so we color it magenta. So there are these 8 kinds of colors, and you can color every compartment in the vesicle with the cargo it contains. Right? So this is something, very, very simple sort of school level math to understand how to label a cell. Then what you do is draw what's called a directed graph. this is the ER, the Golgi, the endosome, and how things move around between them. And then you color them by what ports and cargo they have. And so this is a hypothesis for what a cell is in terms of what cargo are flowing from one place to the other. And this will automatically satisfy what's called homeostasis or steady state by construction. So what we did was to then say, people have been studying how stuff flows around a cell for a long time. But we said, actually the cell is self-organized. The reason this vesicle comes from here to here is not something done from the outside. It's because something about what's here and what's here drives that process. So that turns out to be because the red coat likes the cyan cargo, and I represent that by some sort of interaction. Okay, so this is where I bring the idea of molecules and molecular interactions to the picture. And when I do that, and now I know I'm rushing a little bit, but that's because you don't need to know the details. It turns out that I can take a hypothesis for how a cell works and write it down in terms of what's called a Boolean expression. You have to have this molecule and not that molecule, or this molecule, and it's a big Boolean expression. And then you say, can I find a molecular solution? Can I find a genome that can actually make the cell as I see it? And this is called the Boolean satisfiability problem. So I can write it in some sort of sense with ands and ors and nots. Very, very simple. And then I can start to ask the evolutionary question. Okay? So it turns out that you can actually have many ways to make the same network. Sometimes the same solution can make two different networks. This is called bistability. Sometimes there are two different kinds of networks that are just one mutation away from each other. And over the course of time, these networks can actually change. So now that we have all this math, we decided just to test everything. And so we tested in the computer, and it took a few months, billions of possible cells. Okay, so we just made billions of cells that hypothetically could have existed on Earth. And are they really consistent with how we know molecular interactions work? And it turns out that even though you can write down billions of these networks with nodes and edges and all that, very few of them actually work. It's very difficult to make a cell that actually functions because a lot of things can go wrong. If a molecule does something here, it may screw up something else over there. So if you were just guessing, and if you want an idea of why this is, I don't know how many of you still do Sudoku, but if you did Sudoku by just guessing stuff, you're not going to get it right. There's just too many constraints. This is even harder than that. Okay? So for some reason, it's hard to make, maybe not mysterious, it's just hard to make a vesicle trafficking system. So how do cells become complex if it's so hard to make things? So we brought in the idea of evolution. We said that over the course of time, by mutations, something changes to something else. A simple network can become more complex. And we give some points for complexity. We say that a complex system can have slightly better function, can make the cell slightly more fit. And then we take these hundreds or tens of thousands of networks that we've discovered. These are the networks. Abstractly, each one of those dots there actually represents a network. Each edge over here means that one network can evolve into another. And then you can trace the entire evolution of the network in terms of how many compartments, how many vesicles it has over the course of time. And then, imagine, you can now run the history of eukaryotic evolution on Earth in the computer. And when you do this, it turns out that there's a funneling effect. If you start out from a simple network, over the course of time, you become complex until you reach a stage where you can't go any further. And it turns out with just a handful of molecules that are comparable to what you expect a cell would have had at that time, you get networks that are sufficiently interesting that they do all the things eukaryotes can do today. Phagocytosis, which is where you eat big things, endocytosis and recycling, which is how you signal and sense the environment, and secretion, where you make molecules that go out and, say, enzymatically digest the environment. So this is interesting, right? It's very difficult to make a vesicle trafficking network randomly, but it's almost certain to evolve if you use Darwinian evolution. So evolution is incredibly powerful. It's able to find solutions which you would just not have been able to find by chance. Again, maybe that doesn't surprise those of you who know how evolution works, but so now that we have this tape of evolution running in the lab, we're able to ask questions which nobody's been able to ask before. For example, at some point in time, the eukaryote would have had just the outside membrane and the nucleus, the ER, and it would have got a new compartment like the Golgi from somewhere. So you can ask, did the Golgi come from the plasma membrane or did it come from the ER? And what's the chance of, or something else? What's the chance of one or the other happening? And so over the billions of evolutionary trajectories that we've had, we look for cases where it came from the ER or the PM, and we see how often one happens in the other, and what determines those in terms of what the underlying molecular causes are. So this is where we are now. We're nowhere close to solving the problem, but I think for the first time, we have a plausible evolutionary history of the vesicle trafficking system in eukaryotes. So I'm going to stop now. So this really has echoes of something that's called assembly theory, which is a new kind of framework to understand the evolution of complexity by Lee Cronin and Sarah Walker. The idea is the following. Just mathematically, I can imagine drawing lots of networks, right? And so we call this the universe of possibilities. But then there are physical constraints and chemical constraints, saying these molecules have to be here and those molecules must interact with each other. So the list of possible things is actually smaller than the list of all the things you could write down, just chemically and physically possible. Out of those, there's something that biology also adds, which is called contingency or history. To be able to become something, you have to have a route that gets you there, where you're not dead anywhere along the way. So that historical contingency then restricts the set of possibilities you actually get, even from those that are possible. And eventually you have, the one that you actually see. And somehow you'd like to explain this by putting a series of filters. And I think in some sense that's what we're doing. So that's the end of the talk, except for a couple of slides. So you can read about it in some of these papers. The work is, of course, the work of graduate students and postdocs in the lab over many, many years. And in particular, the wonderful pictures and this vesicle traffic model that we just saw is the PhD work of Sahana. And a lot of this actually got started 15 years ago. And I'll discuss that later when we talk about how I got here. I decided to change what I was working on in the lab, and I found this amazing problem, and I switched over to it. So I'll leave this last slide for the students. So biology is not what we thought it was. Okay? It's not like it says in the textbooks. It's much more interesting. It's much more accepting of outsiders to go work there. You don't have to wear a lab coat and do pipetting, although you can do that also and still make amazing contributions. It's about observation, it's also about theory, it's about computation, it's about data, it's about AI, all that is happening. All this is happening inside biology today. So it's a great place to go. There's a huge ramp ahead, there's a lot of work to be done. And if you don't take my word for it, you can take the word of these very, very smart people. So this is Freeman Dyson, Paul Nurse, Bill Thurston, all amazing physicists, biologists, mathematicians in their own right. So Dyson says, and Dyson, he's a hardcore physicist, right? But he has a view of biology. Already a very old statement, but anyway, he says biology is moving very fast. The line between mathematics and biology is not so sharp anymore. I hope I convinced you. That's where the young people are. They're having the same good times that we had 50 years ago. He's talking about when they invented quantum field theory and how exciting that was. That's how exciting it is today if you do biology. Paul Nurse is a biologist. He got the Nobel Prize for understanding how the cell cycle works. He says the idea, he's a bench biologist. He says the idea is to understand the cell as a logical and computational machine. To shift biology away from the common sense world it is occupied to one that is more abstract. It's an amazing thing for a biologist to say. Right? And it's absolutely true. It's already a 20-year-old sort of call to arms. The final thing is, it is difficult to get into these subjects simply because it's hard to communicate. The language doesn't exist. Yeah? And this is a very nice quote, the famous one from mathematics. This is about math on its own, but I think more generally about how it's hard to communicate between different disciplines in science. Psychological and social dimensions are essential to a model for, let's say, scientific progress. The transfer of understanding from one person to another is not automatic. So venues like this and places where people can work in different disciplines, spend a lot of time understanding each other, developing a common language, articulating questions, just taking the leap and working in interdisciplinary areas. They are rare, but they're increasingly common. And if any of you have the chance to go do something like that at a new interface, you should absolutely do it. You will not regret it. Yes? Sure. Okay, so Q&A. So I didn't have, I already started off with this thing of how I got here. But I hope by seeing the work that we've been doing, you understand, right? We've been trying to find a common language between different disciplines. And this is something where I'm neither an expert on graph theory, nor an expert on cell biology. So at some point in time when you become a scientist, you very quickly find you're no longer an expert on some area where you have to really work. And so you have to just not worry about that. And you have to go and ask people who know better than you and somehow learn from them, right? So I think I've had a lot of mentors over the years who've helped me do that. But I wish I had the opportunity to have something like this, right, where people would come and tell you what they worked on when I was in school or when I was growing up over here. I'm really happy. And by the way, thank you to
to Mount Carmel's and to the Infosys Science Foundation for giving me this opportunity to be here. And thank you all for your attention. And I do want to take questions, anything about this work, about how you do science, how you become a scientist or not. Certainly we're not, we should not all become scientists, but I think we should all know how science works.
Thank you, Dr. Thattai, for elucidating such a complex topic with remarkable clarity. If I may ask a question before we open the floor for the audience, it's a very basic question from the perspective of an educator. A lot of our students, you have highlighted the work of remarkable scientists who've done interdisciplinary work, but a lot of our students come into biological sciences with the hope to leave behind the math and physics they did not like.
Sure.
Some of us educators are also guilty of being very rigid with our boundaries of discipline.
Yeah.
So is there a very organic way of fostering this interdisciplinary work? I understand you can't force someone to make it interdisciplinary.
No. So we don't want to minimize that problem. It is very true that some things people leave behind very early, and that's beyond their control. Okay, so that actually I have a student who joined my lab for a PhD. She did her BSc in biology and eventually ended up liking the kind of models we did so much that she ended up doing a PhD in mathematics, and now she's a professor of mathematics. So some people do that. Also, just to be clear, the kind of math we do, is not so complicated. Right? You can actually, if you like solving puzzles, this is the kind of thing you can pick up if you have an interest in it at any age. And that's why I wanted to show you. There's no deeper mathematics in these little cartoons that I showed you over here. It's not that complicated. What you need is an open mind and not worry about the kind of math that you may have learned in school. The final thing is, we all accept that some of us are better at doing experiments and some of us are better at making models. And nothing prevents you from collaborating. Yeah. So for example, Heisenberg and Einstein didn't know the math that they needed to do their to crack the problems they were working on. Heisenberg didn't know what a matrix was, and Einstein didn't know how to write down these complex geometric equations. So they went out and found mathematicians who they collaborated with. Right? So this is always possible. But the important thing is to realize that there is somebody out there who could help you, and to convince them to come and spend some time talking to you. And we're also busy that really happens. So the reason interdisciplinary work is so hard is because people just don't have the time to, in an open-ended way, learn what people are doing and leave their primary work behind. So if you keep an open mind, it should be possible.
Thank you so much. We'll now open the floor for Q&A. Before we start, I'd like to tell you that I've read somewhere that one question that Professor Thattai dreads is someone asking him what he's trying to cure. So don't ask him that, but ask him anything else.
That's true.
So if you have a question, please raise your hand.
Hello. Thank you for the talk again. I had a question regarding how the cell chooses whether to inherit its property from archaea or the other.
Good question. Cell has no choice or agency in this particular matter. Okay, so evolution works by random changes happening first, and then those changes persist or not because some cells die and some cells survive. The interesting thing is that when you first have the combination of bacteria and archaea, all the bacterial genes, about 5,000 of them, and all the archaeal genes, about 5,000 of them, are in the same cell. The bacterial genes are in the mitochondria, the archaeal genes are in the nucleus. Now, strangely, over the course of time, the bacterial genes from the mitochondria move into the nucleus. Okay? So, a lot of the genes that are only used by the mitochondria today are not in the mitochondrial DNA, they're in the nuclear DNA. And there's a very specific reason for this. So the first thing that happens is the genes flow into the nucleus and only a few get left behind. The second thing that happens is genes get lost. Okay? So if you have two copies of some sort of metabolic gene and you only need one, you keep them both for a while, but if one of them breaks, it doesn't matter. So it goes, and the offspring no longer have it. So there's a process of loss, which is random. And if you did the same thing again, it would be a different subset of genes that persist. The third thing that happens then is duplication. Okay, you have one gene which then becomes 2 and 3 and 4, and each of those subcopies do something different. And these all depend on what's useful for the cell at whatever time and place it finds itself. So now the question you're going to ask, so fine, but then I said there were these patterns that transcription, translation came from archaea, and metabolism came from bacteria. That has to do with the fact that we think in that ancestral state, in the metabolic exchange, the metabolism that the bacteria contributed was very important for the fitness of the cell. And so that's why the bacterial genes disproportionately were the ones that persisted in the metabolic case. But we don't have a full story of this. It's a wonderful open question. It's a great question. And the final question is, how come mitochondria don't lose all their genes? And if you ask me that later, I'll tell you. And why do mitochondria only come from the mother? And a lot of stuff. Anyway.
Two questions. One was that since you said that most of the biochemical genes come from bacteria, why do you believe that there is another set of genes coming from archaea? Why isn't it is an order-wise evolution like how it happened with humans? Like first there was some sort of like chimpanzees, monkeys, like order-wise. Why do you believe that these are? I don't know. I'm just asking.
Yeah.
Because
So
I do not know the basic of it.
No, that's fine. And to be fair, evolution is not taught in schools or colleges to the level it needs to be. It's a very, very complicated discipline. So even I've taken a lot of time to understand all these. So evolution doesn't happen in an order, and in fact, there are many, many examples where the descendants are much simpler and compact than the ancestor. Okay. So these are derived organisms, whereas the ancestor may have been much more complicated. Now, the reason we know that there were bacteria in archaea because we know the sequence of the genomes of the archaea today, and they have all the metabolic genes also, and because they have to survive on their own, and so do the bacteria. So there are two pieces to this. One is a random piece, which is a gene could get lost by chance. The other is a selection piece, where if you lost a gene that was useful, you would die. So first the random event happens, then the selection happens. And that's the way the direction happens in evolution. But there's no necessary direction towards becoming complex. You just become better for your environment. And that could mean becoming simpler. Keeping extra genes for no reason is expensive. And so you tend to lose.
And how long does this simulation?
How long does it take to run? Yeah, each one takes a fraction of a second, but since there were so many networks we had to go over, it took a couple of weeks. But we could make them more efficient. We didn't spend much time optimizing it. In the back there. If you can shout, that's... I can hear you. Yeah. So this is the question you were told not to ask. So what is the implication of this research? Okay. So I think one implication is a very strong demonstration that if you only study a few kinds of organisms, you're not going to fully understand how biology works. Okay? And so by embracing the full diversity of cells that we have on Earth, you'll be able to figure out new and interesting things about how processes function. Now, maybe somebody else can then take the result of that discovery and do something useful. And there are several examples like this. Okay? So there's a group of organisms called ciliates, which are free-living protists. Paramecium is one example. And these ciliates, they don't just have 23 chromosomes like us. They have hundreds of chromosomes. They have 800 chromosomes. And each chromosome contains just a few genes. And because there's so many chromosomes, the ends of the chromosomes, there's many more chromosome ends in Paramecium than there are in us. And they have a name. They're called telomeres. And now it turns out telomeres are part of the key to human aging and the reason why older cells are not as metabolically capable as younger cells and so on. Telomeres were discovered in ciliates. Right? And so people had just stuck to model organisms, we would not have understood that. So there's more than enough examples of people in an open-ended way studying new kinds of organisms. And the results of that over the course of decades coming back to help with human health. But the important thing is that biology is not about human health. And if you're going to try and cure cancer, which is a very laudable goal, you will not do it just by looking at some cancer cell line, right? You will only do it by going and looking more broadly. But I don't do that. I work on the more fundamental questions. Another question.
Good morning.
Sorry, a little louder. Why did I choose to look at vesicle traffic? Yeah. So the way I did this change was when I first moved back to India and I set up my lab. I had a lab where we did something called bacterial genetic networks. Okay, so we took genes, we put them inside cells, and we built what engineers would call positive feedback loops and negative feedback loops, and we compared them to models, and we tried to use them to solve engineering problems. And I thought this was going to be easy, and it turns out to be very, very hard. Almost nothing we did actually worked. And so over the course of those few years when I just set up a lab, I sort of felt that I was maybe had made a mistake, and going from physics to biology is not so easy. And so I took some time in 2010, as I mentioned, to really step back from the work in the lab, find another question. And the way I did this was, there's a place in Santa Barbara called the Kavli Institute for Theoretical Physics where you can run a conference for 3 months. Okay, so and it's nice. It's on the beach. There are blue whales swimming in front of your office and things like that. And so I went, and what I did was I decided I wanted to work on cells and I wanted to work on evolution. And so I literally, instead of reading textbooks, I literally called the experts in cell biology and evolutionary biology over the course of 3 months, and I learned from all of them. And I found that in this interface, a really good open problem where you could make progress reasonably quickly was vesicle traffic, because we knew molecularly how it worked. Whereas other systems, molecularly, you don't know how it works. It's very hard to make progress. So that's how I narrowed down on this problem. It wasn't accidental, but I was lucky that I had the space. Usually you're so busy working on what you're working on, and there's so many pressures to publish and things like that, you don't get to do this. So luckily NCBS is a place where you can do this kind of thing. But I've been then working on that for all this time.
My question is more towards the interdisciplinary side of your lecture. So what challenges do you face guiding the students who are not exactly from your field work?
First of all, no matter what a student's training is, it would not prepare them for this work. Okay, so it's a very good question. When a student joins, they have to be good at something. Okay, they have to be very strong in, let's say, biochemistry, or very strong in, let's say, dynamical systems or calculus, or or programming and analysis. Something. They have to be very strong at something, and so strong at that I don't have to guide them on that particular area. Then you can start to work with anybody and take them forward. Okay, so I think the lesson here is, if you want to do interdisciplinary work later in life, you still have to become very good at some traditional field first, no matter which one it is, and then move on from there. Yeah, and it's not easy, and it's not that my students have an easy time when they come. They have to learn a lot of language, and they typically take a little longer than students who are, let's say, biochemists doing biochemistry to find a PhD problem. But eventually everybody catches up.
So the thing is, I'm studying currently as a chemistry and biotech student. So when would you suggest for me to actually take up research in an interdisciplinary?
You want to do research, right?
Yes.
Yeah. So so on the one hand, you should be really studying chemistry and becoming good at it. But you can start that anytime. You can start that anytime. So by the time I even went to graduate school, I tried 3 different research projects. First of all, to know you may not like research. So forget the field. First you have to just do some research to know if you like it. And it's not for everybody. But if you do like it, then there's nothing like it. So it's a great... But so, the trick is to find a good lab that will host you and give you the space to think and things like that, right? But Bangalore has enough places also that will give you a glimpse of what research will be like. So just look around, and NCBS has a lot of places. Yeah.
Thank you.
There's a question in the front from my mother. So, okay.
Is there anybody working on totally different, like you said, planets somewhere?
It's just somebody working on life on other planets, yeah. It's a discipline called astrobiology. And a lot of it is theoretical, but based on the thermodynamics of how we expect living systems to work, and looking for signatures of imbalance in chemical reactions based on the spectra of planets orbiting distant stars. The James Webb telescope may give us some more data on it. Actually, you should ask Pratik, who would know more about this than me. Yeah, so it's a field called astrobiology, and it's a very exciting field.
If I can add to the question, this is not related to what you spoke about how cells become old and they slow down in terms of their participation in metabolic activities.
Yeah.
But today, because you are also working on evolution, today we are also talking about this whole research happening in reverse aging.
Sure.
So how does that fit?
Yeah, I mean, there's nothing wrong in principle to imagine that the aging process in humans can be modified. Aging is a byproduct of a certain type of life history evolution. So the idea is that after your reproductive age, mutations that would lead to detrimental effects beyond your reproductive age will tend to accumulate in a population, and that's why aging happens. And there are organisms that live much longer than humans, and there are organisms that, for example, mice, for example, don't get heart attacks, and various organisms don't get cancer, and so on. So in principle, there's nothing wrong with exploring those ideas. In practice, it's not going to solve the world's health problems, because whatever therapies one can imagine are going to be very expensive and restricted to a very small group of people who could afford it. Okay, so the question is, why you're doing anti-aging work? Is it just to enable very, very rich people to live slightly longer, like Elon Musk, right? So that's the objection to it. It's not that it can't be done in principle. Yeah, by the way, bacteria also grow old. Yeah, even though they're symmetrically dividing, they grow old. Yeah, so, yeah.
Hi sir, while you were talking about eukaryotes, I was thinking of the red blood cell. And red blood cells do fit the definition of a cell, right?
Sure.
It has a membrane.
Yeah, it doesn't have a nucleus. Very good question.
Yeah.
So you're saying what's going on?
Like what is
Yeah.
where does it fit?
Yeah. So listen. So like what I said right at the beginning, the eukaryote, it's not that it has to have a nucleus. The eukaryote is a phylogenetic and evolutionary definition. All the cells that are descended from this particular ancestral population are all eukaryotes. Okay? So it's not that they have to have a nucleus or not. And by the way, there are a few eukaryotes that don't have mitochondria. They're very rare, and they were only discovered quite recently. You would think that the main job of mitochondria is to do oxidative respiration. That's not the case. There's a huge number of eukaryotes that grow in anoxic environments. They don't need to do respiration, but they still have mitochondria. It turns out that mitochondria are very important for one crucial thing. It's for something called making iron-sulfur clusters, which you need to do other kinds of metabolism. And there's no way to replace that if you lose mitochondria. But there are eukaryotes that don't have mitochondria, there are eukaryotes that don't have a nucleus, red blood cells. Red blood cells are very fascinating cells, because since they don't have a nucleus, they cannot make new proteins, but they still do a lot of things that you expect a cell to do. Good question.
Thank you. So during your talk, I was thinking about the ethical implications of cell engineering or synthetic biology and where they fit in.
Yeah, this is something I used to work on, and it's a very good question. Synthetic biology is extremely powerful. Okay, you can actually take, in any lab with very few resources, and in fact, it's a whole group of people who are called biohackers, who are not necessarily, working in institutions, who are able to modify how cells work. Huge ethical implications. So one is that, of course, cells make copies of themselves. So if you make a cell, it has the potential of getting out of your lab and going out into the wild, and suddenly you have something that didn't arise by the standard biological mechanism. So there's that. And there's the question of, and so whatever the potential byproducts of things getting out of labs. Then there's the question of engineering human cells, and there, ethicists have worked on it. And so there's a very good case to be made that for certain kinds of genetic diseases, it's worth doing sort of stem cell therapies where you take the cell from the patient and modify, put them back in. And there are a few therapies of this type that exist. Where the line becomes very blurry is people try and make these designer babies that will, they're chosen at the time of IVF to have certain genes that will make them taller or run faster or whatever. First of all, none of that is going to work, because we don't actually know how things work to the point where we can select for these things. But again, also the question becomes, these resources are only available to a few people. What exactly is the implication of allowing some people to get designer babies? And so there's a lot of ethical questions over there. But it shouldn't stop research that potentially will allow humans to live more healthily. There are ethicists who work on this, and if you are getting into such a field, there are books about this, and it's a very interesting area to work on.
What were the environmental factors that maybe played a part in this and
What was the environmental factor that played a part in eukaryogenesis? Yeah. This is a completely unknown question, since we don't know the original population from which the endosymbiosis happened. But there are some hints based on what kind of metabolic interactions happen between these cells. I'm not an expert on this, but there's some kinds of chemistry that seem to be present in these archaeal bacterial populations. So that chemistry was important for that environment. Typically, we don't know the environments of ancestral cells, and so we have to ask the reverse question. Based on what we know today about these cells, we can guess something about the ancestral state. It's a tough question. But so I guess another way to ask this question is, did the environment somehow play a role in determining how the subsequent evolution happened? And obviously yes. Obviously yes. Evolution happens in the environment. But the environment is both biotic and abiotic. The real trick for the eukaryote was you had this crazy new cell. It's hard to imagine it was automatically better than the prokaryotes right from the beginning. And somehow it had to beat the prokaryotes back at their own game in existing niches. And so it's very unclear how that happened. And it's likely to be a very difficult process, and that's why eukaryogenesis only happened once. It didn't happen more than once. It's a rare event.
So you talked about the problem of language being sort of like a barrier for education. Do you have any, have you thought of any suggestions on that?
Yeah, I mean, how do you bridge the language barrier? So for example, we have a summer school called Physics of Life for students from physics and chemistry and math and computer science, and we've been running it for 12 years now, where we get students to NCBS for 2 weeks and we expose them to new biology. That's one way. The other way is to try and make sure that any scientific conference always has a diversity of disciplines and doesn't become hyper-specialized, and people are very cognizant of that. But there's a long way to go. So for just very simple things, like when you publish a paper in an interdisciplinary area, when you publish a paper, you have to send it out to something called peer review. You name some experts, and they have to, judge your work. And if you work in an interdisciplinary area, it's very hard to find reviewers who'll be able to even read your paper. And so, I'm not saying that it's easy. So there are lots of little problems, and there are people who are trying to bridge these gaps. Just a couple of months ago, I helped run a symposium in Strasbourg where every talk was given by 2 people. One was from discipline A, and one was from discipline B, and they had to give a tandem talk. Just so that the people in the audience could realize how difficult these things were, because you often only see the final product and not how difficult the whole process was. Yeah, great questions. Yeah, about why they come from.
And where it comes. And you also said someone asked the question how does mitochondrial DNA and cell membrane interact with each other?
Yeah.
So is there any transport vesicles play a role in
No, well, yes. So yes and no. The reason some genes don't go into the nucleus in mitochondria is because proteins that are encoded by those genes, they need to be in such a tight membrane environment in the mitochondria that it's impossible to move them. So the genes that are impossible, the genes whose protein products are impossible to move from the nucleus to the mitochondria, those genes remain in the mitochondria. There's about 10, 20 genes of that type.
I read somewhere, I don't remember, it's a long time, but there is an interaction between paternal genome.
Sure. There's a lot of interactions.
So how is it there or not? I'm not just asking.
It's there. So mitochondria are deeply involved in all kinds of broader cellular processes. And they exchange membranes, they deeply interface. So for example, there's this very important process called apoptosis, which is involved in development of a human. Cells destroy them. Mitochondria are important for that. Right? Just one example. I can keep going with questions if everybody's fine with it. Yeah.
Yes, sir. So, being a physicist originally, what is it that intrigued you to dive into biology?
What was the?
What intrigued you to dive into biology?
Yeah. So actually, if I had known more about biology earlier, I would have got in sooner. So what happened was during graduate school, I took a reading course which was run by a neuroscientist, where they studied a bunch of papers that had been published around 1999-2000, where physicists had been working on biological systems and making a lot of progress in understanding them. And as a graduate student reading these papers, it really seemed like exactly the right thing to do, that we could use these tools that otherwise would have been used to do purely physical systems and apply them to biology. And I think I was right to get into it at that time. In fact, some of the papers I was reading, I did my PhD at MIT in the physics department, and some of these papers that convinced me to get into biology, they were actually written by people at NCBS, Upi Bhalla and Shiv Shankar and so on. So I was reading these papers sitting over there saying, why am I sitting over here? I should be back in Bangalore doing this work, which I am now. Yeah.
So, I have a question. Based on the principle of endosymbiosis, we're saying mitochondria gets rid of all that stuff. But plants have two.
Sure, it happened twice.
It happened twice.
Yeah.
Which according to you is more evolved? Plants or animals?
More evolved, there's no such thing. They're both equally evolved. But you're right, that happened. Endosymbiosis happened multiple times. The plant endosymbiosis happened inside a particular eukaryote, which was the ancestor of all primary photosynthetic organisms. But you know the interesting thing, there are tertiary and quadruple symbiosis. There is a cell that contains another cell, that contains another cell, that contains another cell. At 4 steps down, that also happens. Then there are cells that do something called kleptoplasty, where a cell that doesn't have chloroplasts will go eat a green alga, destroy its membranes, keep its chloroplasts, and do photosynthesis until it divides and one of the cells gets the chloroplast, the other one doesn't, then it goes and eats another one. So there's all kinds of things that can happen.
Simplicity or complexity?
No, so they're both equally complex. I mean, so I really don't. The most complex organisms on Earth, in my opinion, are called ciliates. Tetrahymena, paramecium. They're single cells. They're not multicellular.
I have one more question for you, sir. In brown algae, unicellular
Yeah.
brown algae or green algae,
Yeah.
we have something called as membrane transporters
Yes.
that are very specific.
Yes.
Are the vesicles more evolved?
So in fact, it's amazing. At the molecular level, they operate by exactly the same principles. So the differences really happen at another scale. But inside a cell, if you just went right up to a vesicle, you would not be able to tell if it was from a plant or an animal or an amoeba. They really look the same. It's amazing.
So they are working basically.
Yeah, you can actually take some proteins from there and put it in animals. It will still work. People do that. Okay, I think that's it. Oh, there's another question. Okay. There's one behind you, and then I can go. Yeah. I'll get to that. Wait, fraud question. I just want the person behind to.
My question was, can we think of non-cellular life forms?
Yeah, can you think of non-cellular life forms? So,
And can we see them going to a complex form, like how, if we think about life, we have to
Yeah.
think of cellular forms, membrane forms.
yeah.
And how do we think of non-cellular forms and the chances of their being complex,
Yeah, so
like in other planets?
sure. So I mean, I alluded to this right at the beginning. So I think something like a cell is essential for life, right? So it doesn't have to be a lipid bilayer membrane in this particular size and shape, but the idea of encapsulation and division is probably quite crucial. The only other way to think about it are, totally artificial, sort of silicon-based life-like objects. But I don't really think that counts as life. I mean, living things really go around, are able to find their own resources from the environment, build things, build new cells out of what's available. So I think something like a cell is absolutely essential for any real definition of life. And how do you, I'll take the last question. How do you distinguish between good research and bad research? And this is a very good question. So research, obviously it's hard to put metrics on it, and it's so open-ended that you don't want to score people by saying you're doing good research and bad research. But good research in, let's say, biology or physics or the empirical disciplines, first of all, will always have some way to peg itself against data and the mysteries that are thrown up by observations. So becoming very abstract and just defining your own problems independent of what the data are telling you, that would be bad research. So you need to have some way to anchor yourself in the data. The output of the research is not important, but the way you do the research, which involves some combination of rigor and humility and making sure that you've checked all the options before you make outlandish claims. These are all aspects of good research, which you learn how to do over time. I don't want to say bad research, but sort of poor research is just taking what's known already, changing one thing for no reason at all, reporting what that does, because that's a waste of resources. You're doing this, but without some bigger picture in mind. Simply, just to carry on what looks like research. I think I won't say anything more than that, but it's a very good question, and I think if you're going to become a researcher, you'll develop your own taste over time on what seems to be working for you or not, and hopefully your mentors will be able to guide you in answering that question. Okay, great. Thank you so much.
Thank you, Dr. Thattai. May I request you to take a seat amongst the members of the audience as you've been standing for a while now?
Sure.
And I will quickly wrap up the event with a word. Delighted to have Professor Thattai's parents here with us today. So, on behalf of Mount Carmel, we give away a small token of our appreciation to the veterans. Ladies and gentlemen, as we conclude with this lecture, it is my honor to extend thanks to all those who've made this event possible. We express our gratitude to the Infosys Science Foundation for entrusting us with the opportunity to host Professor Thattai's lecture. His insightful research has sparked valuable discussion and broadened our understanding, and we are proud to have facilitated this exchange of ideas. We also extend our thanks to Professor Thattai for sharing his groundbreaking work with us today, and mainly for simplifying such a complex topic for us. A special acknowledgement goes to Bhavna Mehra, the head of Infosys Foundation, who is an esteemed alumna of MCC. It is a pleasure to have you back and to have your leadership and support for this event. A special thanks to the management of MCC and to the Department of Communication Studies, whose efforts have made this event possible. We are also grateful to the audience for your enthusiastic participation and engagement on a Saturday morning at 9 a.m. Thank you once again for being a part of this lecture. Looking forward to more such interactions. Have a good weekend.